glycoproteins. Okay. All right. Biological importance. We have two important processes. We have glycosylation and glycation. Now, this has got to do with addition of sugar into a protein. And so the protein now has sugar attached to it, and they are called glycoproteins. Okay, so glycoproteins are proteins that contain oligosaccharide chains covalently bound to the amino acids. All right, so glycosylation. This glycosylation is the attachment of sugar utilizing an enzyme. So it is an enzymatic reaction. Okay, is the most frequent post-translational modification. What we mean by post-translational modification is when a protein that has been synthesized, then it is modified by addition of other compounds. Now, in this case, the compounds that are added are sugars. So that's why it's called glycoproteins. The process is glycosylation. It is utilizing enzymes for the attachment of sugar to the protein. While the other one is glycation. Glycation is the attachment of your sugar to the protein without the use of enzymes. A very good example of this is in diabetes mellitus, where there is a very high sugar concentration in the blood. The sugar reacts without any enzyme, reacts with hemoglobin. And so the hemoglobin is decorated with glucose, because glucose is the sugar in the blood. Okay, so it's decorated with glucose. The midtext knows about this. This is what they call hemoglobin A1C or glycated hemoglobin, when the blood sugar increases in amount than normal. Okay, so that's glycation. And it says that this process have serious pathologic consequences. Example was the one that I mentioned, uncontrolled diabetes mellitus. Okay, so glycoproteins are class of glycoconjugate or complex of carbohydrate. Complex carbohydrate. Molecules contain one or more carbohydrate chains linked to the protein covalently to form glycoproteins, or sometimes it is also called proteoglycans. Okay, or the sugar is attached to a lipid, so the lipid becomes known as glycolipid. Almost all plasma proteins, that's your topic for tomorrow, no? And many peptide hormones like glycoproteins, and also the blood group substances of the RBC, you know, it's the blood group, ABO. No? Others are glycosphingolipids. Okay. Now, cell membranes as well contain substantial amounts of carbohydrate attached to the protein on the lipid bilayer. So there are so many reports now that alterations in the structure of glycoproteins and other glycoconjugates of on the surface of cancer cells are very important in the metastatic process performed by the cancer cells. Okay? So it says glycoproteins occur widely and perform numerous functions. Now, the amount of carbohydrate that is attached to the protein varies from 1% to 85% by weight. So it varies, no? From 1% to about 85% by weight of the glycoprotein. So it says also, it's here says, the structure of the glycoprotein changes in response to signals involved in the differentiation of cells. In normal physiology, as well as in neoplastic transformation. When I say neoplastic, example is your cancer cells. Neoplastic transformation. This is because of the different way by which your glycoside transferases work. This enzyme glycoside transferases are responsible for the attachment of the sugar into the protein in glycosylation. Remember, we said glycosylation, it is enzymic, meaning enzymes are involved, and the enzymes are glycoside transferases. So let's look at table 46-2, and let us look at the major functions. Okay, function, the glycoproteins. So you have here function and glycoproteins. So it can act as a structural molecule. This is brought about by collagens, lubricant and protective agent, mucin. Now this mucin is very, very important, no, because it says protective agent. Now your mucus, there's only a small amount of about 5% of mucins are in there, but that is quite important because that's mucins are the substance that make the mucus into a gel-like substance. Okay? Now, mucins also as a protective agent. Now, you know that your stomach secretes a powerful acid, hydrochloric acid, and a protease or a protein enzyme that digests the protein, and that is pepsin. And then you are just wondering why the stomach is not being acted upon by the acid and by the enzyme protease, since the inside of your stomach is a protein. This is because of the mucin that covers it. Without that mucin, it will be eaten up. And that's why we have ulcers. No? Now, every time you smoke, the production of mucins is inhibited. So there is a relationship between smoking and gastric ulcers. It's because of that. All right. So these glycoproteins also transport molecule. So transferrin, which transport iron, and ceruloplasmin, which also transports copper. And then you have immunological molecules. You have immunoglobulins, you have histocompatibility antigens. These are the ones that are found in your surface of your RBC. And that's why you will have several blood groups, the ABO. Hormones. Now, chorionic gonadotropin, your thyroid stimulating hormone, are glycoproteins. And then enzymes that are glycoproteins, there are many. An example is alkaline phosphatase. Okay. Then cell attachment recognition site. This is very, very important in fertilization, where the sperm is going to attack the egg cell or the oocyte. Now, this is also important in attachment of virus to the cell, just like the coronavirus attaching to our normal cells and destroying it. Or bacteria that attaches to the cell. Or signal proteins or signal glycoproteins. Example, your hormone, which when attached to the hormone to the cell, then interactions occur. And then antifreeze. This is plasma proteins of the cold water fish. So you just be wondering why the fish that are found in the North Pole or the South Pole, where the water is very, very cold, why don't they get frozen? They're living under the ice. No, the polar ice caps. They're underwater. Okay. So the glycoprotein acts as antifreeze. Okay. And also, you'll be wondering why their fat will not harden. You have seen that your fat, anything that is fatty or oily, you place it in a refrigerator, they're always freezing of the oil or the fat. Diba? You notice that? No? But it does not happen to the fish because there are certain glycoproteins that acts as an antifreeze so that the blood will not be frozen. And not only that, not only the glycoproteins, but also the unsaturated fatty acids, because the freezing point of your unsaturated fatty acid is quite very, very low. No, very low temperature. All right. Continuation, we have interact with specific carbohydrates, lectins. Now, these lectins, these are proteins that be able to attach to the carbohydrate, lectins, as well as selectins. And then, of course, antibodies, be able to recognize the specific carbohydrate on the surface of the protein. Then, glycoproteins are also used as or forms receptors. Cell surface proteins involved in hormone and drug action. And then we have proteins that regulate folding, regulate the folding of proteins. We call them calnexin, or the other one is calreticulin. They belong to what you call chaperone proteins. These chaperone proteins aids in the folding of the protein molecule after it is being synthesized. So if there is a mistake in the folding of the proteins, the same chaperone proteins will prevent it from excreting out of the cells where they were formed, so that they will be degraded back into amino acids. Okay. The regulation of differentiation and development. NATCH and its analogs K protein and development. And then hemostasis. Okay. And thrombosis. Specific glycoproteins on the surface membranes of platelets is needed for hemostasis or blood clotting. Okay? So that's your function of your glycoproteins. Now, the oligosaccharides or the carbohydrate or the sugar chain attached to your protein in the glycoproteins have some functions. So they are there. So it changes physiochemical properties of the protein, such as solubility. You know, sugar is soluble in water. So the more sugar attached to the protein, it makes the protein more soluble. And it also increases the viscosity. Okay? Viscosity. Now, kung women who are cooking knows that they use starch, no, in order to make the soap more viscous. So they add starch. And starch, of course, is made up of glucose. As well as it changes the charge of the glycoprotein. Supposing the sugar attached, like for example, sugar acids. Now, sugar acids like glucuronic acid. So it is positively charged. See? So it also determines the conformation of the molecule. The chain of sugars or oligosaccharides. And then denaturation, meaning the nature of the protein has been demolished. It is denaturized. Okay. Another function is provide binding sites for various molecules, as well as bacteria, viruses, and parasites. It also provides cell surface recognition signals. Cell surface recognition signals. Okay. And it protect against proteolysis. Remember the mucins that I mentioned that protect the surface, the inner surface of your stomach from the effects of the proteolytic activity of pepsin, no? And also the hydrochloric acid. So it protect against proteolysis. So the protein it could not be lysed because of the presence of this oligosaccharides or a chain of uh, sugars attached to the protein. It also ensures correct folding. Remember, I mentioned your calnixin for the protein folding that are exported from the cell and target incorrectly folded proteins for transport from the endoplasmic reticulum back to the cytosol for degradation. So if it is incorrectly folded proteins, then we also remove that. It is not allowed to be transported outside of the cell and they have to be degraded in the cytoplasm. This also protect peptide hormones and other plasma proteins against the clearance by the liver. You know, the liver does a lot of things. It is the center of, remember our topics of xenobiotics. So hormones and other proteins that passes to the liver would be degraded, but it has to be protected. Like for example, hormones. So protect the peptide hormones and other plasma proteins against the activity of the liver. Then we have it allows anchoring of extracellular proteins in the cell membrane and of intracellular proteins inside subcellular organelles such as endoplasmic reticulum and the Golgi apparatus. Then it is also involved in direct intracellular migration. So if these oligosaccharides that is found in cancer cells on the surface of the cells, then migration can occur. And migration of cancer cells from the nidus or the nest of the cancer cell is known as metastasis. So this is how it is, how they metastasize through the help of your oligosaccharide chains of your glycoproteins. Affects embryonic development and cell and tissue differentiation. So some tissues could not be developed because of the absence of certain oligosaccharide chains of your glycoproteins. Oh, here is the one now. May affect sites of metastasis selected by the cancer cells. Okay. The oligosaccharide chain has so many functions. We mentioned that. And it encodes biologic information. The biologic information and the sequence and linkages of sugars in glycans differ from that of the biologic information furnished by the DNA and RNA. Now, this biologic information is secondary only, no, rather than primary information. Primary information from the DNA and RNA. The one that is rendered by the oligosaccharides is secondary. So the pattern of glycosylation, so again, remember the word glycosylation, it requires enzyme, no, for a sugar to attach to a protein molecule. And the enzyme, so many of them, and they are named glycosyl transferases. Glycosyl. So glyco meaning sugar, no, transferases. So they are involved in glycoprotein synthesis. The information from sugar, it can be detected when it reacts with an important compound known as lectins here. Okay. All right. Eight sugars are involved in glycosylation of your glycoproteins. And what we have, table 46-3, principal sugars found in human glycoprotein. Okay, the sugar, the type of sugar, abbreviation of the sugar, and then the carrier, the nucleotide, and you have comments here. Galactose is a hexose, abbreviation GAL. Carrier is iodine dinucleotide phosphate, UDP galactose. Okay. Often found at the subterminal to neuraminic acid link, nitrogen linked glycoproteins. Also found in the core trisaccharide of proteoglycans. That's where their location. Another sugar, glucose. And you have, and the type is a hexose, just like your galactose. It is actually a isomer of glucose. Galactose is isomer of glucose, as well as mannose is also an isomer of glucose. No? They are all hexoses. Now, all the sugars, we know that our blood sugar is glucose. Okay? Now, this glucose can be transformed into galactose, or this glucose can be transformed into mannose. Right. Because you might be wondering why we know our blood glucose is our blood sugar is glucose, and then why do we have this galactose and this mannose? These are isomers of your glucose. We can easily transform that through your isomerase. Okay. So you have glucose. Its carrier is UDP glucose, present during the biosynthesis of nitrogen-linked glycoprotein, but usually, not usually, present in mature glycoproteins. They are also present in some clotting factors. Okay. Mannose. Okay. You have here GDP. Here, for glucose, it is UDP glucose, uridine diphosphate, the nucleotide that is attached to that glucose. Now, for mannose, it is GDP, guanosine diphosphate. Now, they have different nucleotide. Galactose, UDP. No? This one is UDP. This one is GDP. Okay. A common sugar in nitrogen-linked glycoproteins. Then you have N-acetylnuraminic acid. Type, it is a sialic acid. 9 carbon atoms. Okay. So this is the abbreviation, N-acetylnuraminic acid. And the nucleotide carrier is CMP, cytosine monophosphate. Often seen as a terminal sugar in both nitrogen and oxygen-linked glycoproteins. So they are found at the ends. Okay, other types of sialic acid are also found, but the nuraminic acid, N-acetylnuraminic acid, is the major species found in humans. Now, the acetyl groups can also occur as oxygen link. No? Instead of nitrogen, it is oxygen link species. Okay. Then we have fucose. Fucose is a deoxyhexose from glucose. From glucose, your glucose loses a hydrogen. Loses an oxygen, I'm sorry. And the abbreviation is FUC. Carrier nucleotide GDP. May be external in both nitrogen and oxygen linked glycoproteins. Okay, we'll show you later on this nitrogen and oxygen link. Then you have N-acetyl galactosamine, the amine of galactose. It is now type is amino hexose. Abbreviation GalNAc. Carrier nucleotide UDP. Present in both nitrogen and oxygen linked glycoproteins. Then we have N-acetyl glucosamine. This is the amide form of your glucose, acetylated. So it belongs to the same category, amino hexose. So you have here GLC-NAC. So similarly, you have UDP as its nucleotide carrier. Sugar attached to the polypeptide chain via asparagine link, nitrogen link, asparagine. Oxygen link, usually it is serine or threonine. Okay, then we have xylose, which is a 5-carbon atom sugar. It's a pentose. And carrier is UDP. Attached to the OH of serine. And in many proteoglycans. Xylose, in turn, is attached to 2 galactose residues, forming a link of trisaccharide. All right. Let's look at this before going to that. Okay. This is your sugar. This portion here is the protein. And this is serine. It's supposed to be an OH. That's why it's carbon to oxygen link. So when you say this is oxygen link. Now this one is again the sugar. This is the protein. This is asparagine, carbon to nitrogen. You see that? Carbon to nitrogen. And then this one, it is also glycine, the amino acid, nitrogen, with an ethanolamine before it is attached to mannose. Okay? So that is nitrogen link. This is oxygen link. Okay. So you have oxygen linkage and acetyl galactosamine to serine. Nitrogen link and acetyl glucosamine to asparagine. And the other one, glycosyl phosphatidyl inositol, GPI. GPI. That's the one. GPI means glycosyl phosphatidyl inositol. Okay, so you know that. Now, lectins can be used to purify glycoproteins to investigate their functions. So your lectins, because we said the lectins can attach to the carbohydrate. So lectins are carbohydrate binding proteins that agglutinate cells or precipitate glycoconjugates. A number of lectins are themselves are glycoproteins. Now, immunoglobulins that react with sugar are not considered to be lectins. Lectins contain at least 2 sugar binding sites. Proteins with only a single sugar binding site cannot agglutinate cells or precipitate glycoconjugates. Lectins are first discovered in plants and microorganisms. But many lectins of animal origin are now known, including the mammalian asayalo glycoprotein receptors. Many peptide hormones and most plasma proteins are glycoproteins. Treatment of protein with neuramidase removes the terminal neuraminic acid. Okay. This asayaloglycoprotein is cleared from circulation faster than the intact glycoprotein. So they are removed right away. Liver cells contain asialoglycoprotein receptors that recognize galactose moiety. So the galactose that is attached there of the many desialylated, meaning proteins in which the sialic acid has been removed. And then they are destroyed. So they undergo endocytosis and catabolism. So they are taken in by the cell and they are catabolized. Plant lectins are formally called phyto. Phyto for plants. Hema, that's heme, glutinins, because they are able to agglutinate RBC by reacting to the cell surface glycoproteins of the cell membrane of your red blood cell. Now you will find undenatured lectins in your food. No? These are when legumes are undercooked. So they're not, the structure is not removed. So what does it do? It strips the intestinal mucosa, I'm sorry. It strips the intestinal mucosa by agglutinating the mucosal cells. See? So be careful with uncooked legumes. You know what are legumes? No? Beans. So they have to be thoroughly cooked in order to denaturize your lectins. Lectins are used to purify glycoproteins as tools for probing or looking into the profile of your glycoprotein of the cell surfaces and as reagents for generating mutant cells deficient in certain enzymes. Okay. So that's the use of lectins. Lectins can be used to purify glycoproteins to investigate their functions because they can attach to the glycoprotein. So we said glycoproteins can be divided into three main groups based on the nature of linkage, no, between the polypeptide and the oligosaccharide. You remember that? All oxygen link, nitrogen link, and GPI link. So there you are. Oxygen link, nitrogen link, GPI link. So this is the protein as glycine there. Okay. So these are the three. Those containing oxygen glycosidic linkage and those containing nitrogen glycosidic linkage and those linked to the carboxyl terminal amino acid of proteins. These are anchored to your glycosyl phosphatidylinositol anchor, GPI anchored proteins. Okay. Glycoproteins contains several types of glycosidic linkages. For oxygen link, we usually have this serine and threonine. No? Now, here we have N-acetyl galactosamine, which is a sugar attached to serine and threonine. It's the predominant linkage, usually galactose, or an acetylated muraminic acid attached to the acetyl galactosamine. This type of linkage is found in mucin. You remember that mucin? Then proteoglycans contain a galactose to galactose xylose trisaccharide, the so-called linked trisaccharide attached to serine. And then collagens, you have galactose attached to your hydroxylysine, hydroxylated lysine. Very, very important in collagen. Many nuclear and cytosolic proteins contain side chains consisted of single nitrogen acetyl glucosamine attached to serine or threonine. Okay. Mucins have a high content of oxygen-linked oligosaccharides and exhibit repeating amino acid sequence. Because of this, high content of oxygen-linked oligosaccharides and repeating amino acid sequences, this would be very, very difficult for proteases to act on the protein portion, the polypeptide chain. So, they help to lubricate and form protective physical barrier on epithelial surfaces like the inner surface of the stomach and intestine. Now remember that enzymes that goes into the stomach and intestines are very powerful enzymes that they can digest the protein. It's a proteases. Mucins have two distinctive characteristics: a high content of oxygen-linked oligosaccharides, generally more than 50%, and the presence of variable numbers of tandem repeats of peptide sequence or VNTRs. So what do you mean by this? Variable numbers of tandem repeats, meaning of the same amino acid sequence. Several of these, to which the oxygen glycan chains are attached in clusters. This tandem repeat are rich in serine, threonine, and proline. Up to 60% of the dietary requirement of threonine can be accounted by synthesis of mucins. So imagine that. So what is so distinctive feature of serine is the OH. Serine and threonine have OH, and proline can be hydroxylated to become hydroxyproline. So there are both secretory, or meaning secreted out, and membrane-bound mucins. Mucose secreted by the gastrointestinal tract, the respiratory tract, reproductive tracts, has about 5% mucin. So that's mucose, no? Where's that now? Mucose has a high viscosity and forms gel because of mucin content. The high content of oxygen glycans confers an extended structure. It explains the steric interaction. All right. Membrane-bound proteins participate in cell-to-cell interaction and mask cell surfaces antigens. Many cancer cells form large amount of mucins. Many cancer cells form large amount of mucins that mask the surface antigen. So therefore, your antibodies cannot recognize them. No? It protects the cancer cells from immune surveillance because it is being masked by mucin. I say many cancer cells form large amount of mucins to cover the surface antigens of that cancer cells. So it cannot be acted by your immunoglobulins. Mucin also carry cancer-specific peptide and carbohydrate epitopes. Some of this has been used to stimulate an immune response against cancer cells. So once you remove that epitopes, then that can be used to stimulate your immune system to produce more antibodies. Oxygen-linked glycoproteins are synthesized by sequential, one after the other. One after the other, addition of sugars from the sugar nucleotides. So when you say sugar nucleotides, your UDP glucose, UDP galactose, no? Your CMP mannose. Okay. So those are sugar nucleotides. So they add one sugar after another after the sugar nucleotides. So because the glycoproteins, because most glycoproteins are membrane bound or secreted, their messenger RNA is usually translated on the membrane-bound ribosomes. So the glycan chain or the glucose chain or whatever sugar chain are built up in sequential donation of sugars from sugar nucleotide. Remember UDP glucose, UDP galactose, no? Okay. Catalyzed by glycoprotein glycosyltransferase. Because many glycosylation reactions occur within the lumen of the Golgi apparatus, there are carrier proteins. And what are these carrier proteins? They are the permeases. So from the word permeases or permeate, okay? And transporters, which transports. To transport sugar to nucleotides, UDP galactose, GDP mannose, CMP, acetylneurominic acid, across the Golgi apparatus, in the Golgi membrane. They are anti-porter systems. Influx of one molecule of sugar is balanced by the efflux of one molecule corresponding nucleotide, UMP, GMP, or CMP. Okay. There are 41 different types of this glycosyltransferase. So there are many glycosyltransferase. The end-linked glycoproteins contain an asparagine and acetylglucosamine linkage. The nitrogen-linked glycoproteins are the major classes of glycoproteins, including both membrane-bound, so that is attached to the membranes of the cell, and the circulating glycoproteins. So they are distinguished by the presence of asparagine and acetylglucosamine linkage, shown in figure 46-1. There are three major classes of this nitrogen link oligosaccharides. We have complex, we have high mannose, and hybrid. All three classes have the same branch pentasaccharides found to asparagine but differ in their outer branches. Let's show that. Figure 46-2. Oh, here we are. Okay. If you look at this, you have the amino acid asparagine. And then you have N-acetylated glucose, two molecules, two molecules, two molecules. And then you have mannose, three molecules, then three molecules, then three molecules. These three, complex, hybrid, and high mannose, are similar only up to there. Only up to there. So they are similar. They differ in the terminal portions. Like the complex contains, as we see here, you have your glucose NAC, okay, similar to that. Then galactose, then sialic acid. Sialic acid, your neuraminic acid here. While this one, hybrid, it looks similar to that because of this, but it has mannose. No? And then galactose, similar to that. So it's a hybrid. While this one is distinct, it is highly mannose as the sugar. There. They are all mannose. Okay? So you have three types: complex, hybrid, high mannose. So mostly mannose. Complex oligosaccharides contain 2, 3, 4, or 5 outer branches as the complex. The oligosaccharide branches are often referred to as antennae or antenna. So antennae. You have 2 antennae, it's called biantennae. 3 triantennae, tetraantennae, and pentaantennae. No? Ah. So we have pentaantennary structures may be found. They generally contain the terminal N-acetylnuraminic acid residues. And you have the similar, the similar, remember the similar? Okay. 3 mannose here, 3 mannose here also, 3 mannose here. 2 gallnuts, 2 gallnuts, 2 gallnuts. Similar, but they differ in the terminal portion. Now you have here 1, 2, 3 branches, so that is triantennae. Also this one, 1, 2, 3, triantennae, and then diantennae. Okay. So there are complex type which ends in galactose or fructose. We said that it usually ends in uraminic acid. No? They generally contain terminal N-acetylnuraminic acid. The complex may end in galactose or fructose. Now, these high mannose oligosaccharides have 2 to 6 additional mannose residues linked to the pentasaccharide core. What's the pentasaccharide core? So remember, this is your pentasaccharide core. Here. 5 saccharide molecules. 1, 2, 3, 4, 5. Okay? Now, dolicol the biosynthesis of end-linked glycoproteins involves dolicol. We will be able to see this compound when we take up metabolism of glucose. Oh. Okay. So this is the structure of dolicol. Okay, so it's a very big structure, maybe about 90 to 95 carbon atoms. Now this is common. This is what we call isoprene unit. Okay. And the dolicol here, this isoprene unit, which is 5 carbons. Now N is 17 to 20 isoprenoid units. So this is isoprene unit. You will also meet this in the synthesis of cholesterol, isoprene units. Okay. Involves dolicol, pyrophosphate, oligosaccharide. So all nitrogen-linked glycoproteins begin with the synthesis of branch oligosaccharide attached to dolicol pyrophosphate on the cytosolic side of the endoplasmic reticulum, which is then transferred or translocated to the lumen of the endoplasmic reticulum, where it undergoes further addition of glucose or sugar glycosylation before the oligosaccharide chain is transferred by your transferase into asparagine residue of the protein, the apoprotein. Okay? As it enters the endoplasmic reticulum during the synthesis of membrane-bound polyribosomes. So the synthesis is brought about by the ribosome, which is located on the surface of your glycoprotein. I mean, the surface of your Golgi apparatus. Okay, so you have here. So many of sugars added. So it is here dolicol pyrophosphate, there, no? Added. So those sugars will added now to dolicol pyrophosphate, coming bigger. So you have your dolicol here. So you have now a sugar that is added M, if of course mannose, no? You know that. Then here is another one, addition, some more. So there were already 2 of this, and then 6 of mannose. All right. So until finally it is this. So starting from your dolicol pyrophosphate. This is quite a big molecule, this dolicol alone itself. Proteins, glycoproteins, and calnixin ensure correct folding. So remember when you said this, calnixin, oh, here it is. Calnixin is a chaperone protein. And the function of chaperone protein is to ensure correct folding of the protein. Otherwise, if it is incorrectly folded, it is not excreted out, but goes into the cytoplasm and then it is destroyed there in the cytoplasm back into amino acids. Calnixin binds to glycoproteins that possess monoglycosylated core. Monoglycosylated core. Meaning only one type of sugar, in which the terminal glucose residue is removed, leaving only innermost glucose attached, and calnixin bound to a glycoprotein form a complex with ARP57, a homolog protein of an isomerase, a disulfide isomerase, which catalyzes disulfide bond interchange, and facilitating correct folding. You know, your disulfide bond is one of the covalent bonds. Uh, like your peptide linkage or your glycosidic linkage, they are covalent bonds. So if it is correctly folded, it is available for secretion outside the endoplasmic reticulum. If not, a glucosyl transferase recognized and rebinds the calnixin. It is now correctly folded. There is another protein which is similar to the function of calnixin, and that is your calreticulin. Performs similar function to that of calnixin. So there are several factors that regulate the glycosylation of glycoproteins. Now, once there is a mistake, then that is not secreted out. It is thrown into the cytoplasm so that it will be degraded. Now, cancer cells forms a different pattern of glycosyl transferase from those in normal cells. As a result, specific gene activation or repression now occurs. The differences in oligosaccharide chain could affect adhesive interactions between cancer cells and normal parent cells, contributing to metastasis. So they depart from the normal cells. If they cannot depart from the normal cells, then nothing happened to that cancer cells. It will just die down. And cancer cells feeds on glucose. Always remember that. It feeds on glucose. Because cancer cells utilizes anaerobic glycolysis as a way of producing ATP for its own use. So advise cancer patients not to take in large amount of carbohydrates because it will be converted to glucose. So you can starve the cancer cells by doing that. So you need to starve the cancer cells. Now, the GPI anchor allows mobility of the protein, your glycoprotein, on the plasma membrane. Enhance the mobility. They are attached to the outer leaflet of the lipid bilayer. Attached to the outer leaflet. So they are free to diffuse than proteins that are anchored through both layers of the membrane, which we call integral proteins. Increased mobility is important in facilitating rapid response to stimuli. The GPI anchors may connect with signal transduction pathways. Signal transduction could be brought about by hormones or brought about by other signal molecules like cyclic AMP. So there are receptors for hormones and other cell surface signals. Cell surface signals. So that tells you, like for example, in hypoglycemia, the glucose concentration is quite low. So there is no longer so much glucose diffusing into the cells. Now, these glycoproteins can sense that there is less glucose. So stimulation of the formation of glucose through process known as gluconeogenesis can be brought about by these signal transduction pathways, which is, of course, through the glycoproteins for signal transduction. Okay. All right. Some proteins undergo rapidly reversible glycosylation. Now, glycosylation is addition of sugar to the protein without the help of any enzyme. So this is just brought about by an increase of glucose concentration so that the protein is glycosylated. So we have glycylation. Advanced glycation end products or AGEs are important in causing tissue damage in diabetes mellitus. So it says here, AGEs for advanced glycation end products, important in causing tissue damage in diabetes mellitus. Glycation, addition of glucose without the use of enzyme. Okay. This can bring about browning of certain foodstuffs during storage or heating and provides much of the flavor of some foods by rearrangement of the protein. We call it amaduri, rearrangement. Now, this is subjected to heat. Okay, and further reactions to yield advanced glycation end products. The overall series of reactions is known as Maillard reaction. Maillard reaction. It's involved in the browning of certain foodstuffs during storage or heating and provide much of the flavor of some foods. Okay, so this is your protein molecule, determined amino acid. Here is glucose, non-enzymic, and it reacts with the amino group here, with the aldehyde group. There you are. And so you have that linkage. That linkage is CONH linkage. Huh? CONH linkage. Yeah, rearrangement by Amadore rearrangement. Then you form amino ketone, advanced glycation end product. Advanced glycation end products underlie tissue damage and poorly controlled diabetes mellitus or uncontrolled diabetes mellitus. When the blood glucose concentration is very high, there is an increase addition of sugar to the protein. Glycation of collagen and other proteins in the extracellular matrix alters their properties, increasing cross-linking of collagen. Okay? So what happens if you increase the cross-linking of collagen? The blood vessels become stiff. And it leads to various accumulations on the blood vessel wall, various plasma proteins, LDL, and as a result, you can now form atherosclerosis or atherogenesis, the formation of atherosclerosis. Ages appear not only in on the small blood vessel, microvascular, as well as macrovascular. Now, in elderly persons, if you look at the X-ray of the heart, the arc of the aorta, you can see deposition, no, on the intima side of the blood vessel of the aorta. So there is accumulation of various plasma proteins there. And notable is your LDL or your low density lipoproteins. So that's brought about by that glycation. Another is hemoglobin in your red blood cell leads to the formation of, here we are, here, hemoglobin A1C. Now, this is really a very good marker for an increase of blood glucose. Very good marker. Hemoglobin A1C. You may not have the signs and symptoms yet of your diabetes mellitus, but if they take a blood sample and look for the hemoglobin A1C. And if there are hemoglobin A1C, even if you do not have the signs symptoms of diabetes, you already have now what they call diabetes mellitus. Okay. Now, it's like this. You form ages, and the endothelial cells and macrophages have age receptors on their surfaces. So uptake of glycated proteins by these receptors can activate transcription factor NF-kB, generating a variety of cytokines and pro-inflammatory molecules. Okay, so there is an inflammatory process due to the cytokines and inflammatory molecules. So hemoglobin A1C occurs normally to a small extent. A normal person, there's a little very amount, sometimes undetectable, but they are common in pre-diabetic or frank diabetic patients with poor glycemic control, whose blood glucose concentration is consistently elevated. So the measurement of hemoglobin A1C is used for management of patients with diabetes mellitus. Glycoproteins are also involved in many biological processes and many diseases, as listed in your figure 46, your table 46-1, you have the different functions. Excuse me. They have different functions, and if there is something wrong with the glycoproteins, then you may have problems with that function. Okay. They are also important in fertilization and inflammation due to defects in the synthesis and the breakdown of glycoproteins. Here we are. Glycoproteins are important in fertilization. In your introduction to genetics, in your previous studies, no, the egg cell or the oocyte is covered with known as the zona pellucida. Zona pellucida. So the sperm, in order to fertilize the egg cell, they have to go through the zona pellucida. So it is a thick, transparent envelope that surrounds the oocyte or egg cell. It is a glycoprotein, and it is known as ZP3, zona pellucida 3, an oxygen-linked glycoprotein that functions as a sperm receptor. A protein on the sperm surface interacts with the oligosaccharide chain of CP3 by transmembrane signaling, and it induces acrosomal reaction. Now that acrosome is on the head of the sperm cell. That acrosome contains enzymes that would digest the zona pellucida. Okay. So what are these enzymes there? Proteases and hyaluronidase. Hyaluronidase. And other contents of the acrosome or the head of the sperm are released. These enzymes, as it eats up the zona pellucida, the sperm now can pass through the zona pellucida and reach the plasma membrane of the egg cell. Another glycoprotein, PH30, is important in the binding of the sperm plasma membrane so that the oocyte and subsequent fusion of the two membranes, enabling the sperm to enter and fertilize the oocyte. So, what does this tell us? It tells us that the egg cell is protected by zona pellucida made up of glycoproteins. And the head of the sperm cell or the acrosome contains enzymes to digest the zona pellucida. And it contains a glycoprotein so that once the sperm passes through the zona pellucida and hits the membrane of the egg cell or oocyte, you have, excuse me, you have a glycoprotein PH30 important in the binding of the sperm membrane to that of the egg cell, so that the two membranes will fuse together so that now the sperm can enter and fertilize the egg cell. Now, sometimes the zona pellucida of the egg cell can be altered if there is a wrong synthesis of the oligoproteins. And the sperm, which carries the enzymes to digest the zona pellucida, might not be able to digest the zona pellucida. And so there is no fertilization that can occur. And also sometimes The enzymes covered, brought about by the sperm cell on its acrosome, let us say that there is something wrong with the enzymes that it carries, so it cannot also digest the zona pellucida. So therefore, there is no fertilization that can occur. Okay. All right. Silictins are surface lectins, and they have role in inflammation and also for the lymphocyte to be able to target, you know, the cell where this should function. Leukocytes play important roles in many inflammatory and immunological processes. The first steps are interaction between leukocytes and endothelial cells in circulation prior to the passage of leukocytes out of the circulation. Leukocytes and endothelial cells contain cell surface lectins, and these cell surface lectins are called silictins, which participate in intercellular adhesions. Silictins are single chain calcium binding transmembrane protein, and the terminal contain lectin domains, which is involved in binding to specific carbohydrate ligand sites. Interaction between silictins and neutrophil cell surface and glycoproteins on the endothelial cell trap the neutrophils temporarily so that they roll over the intercellular endothelial surface. And doing so, the neutrophils are activated and change in shape and adhere firmly to the endothelium. This adhesion results as an interaction between integrins Okay, let's continue. So after adhesion, the neutrophils insert pseudopodia into the junctions between endothelial cells and squeeze through these junctions and cross the basement membrane, and they are free to migrate to the extravascular space. Excuse lang kay very itchy throat. Lectins bind sialylated and fucosylated oligosaccharides. So lectins bind to compounds in which sialic acid is attached and fucose sugar is also attached. No? As well as sulfated lipids, the lectins can attach. Now, cancer cells have selectin ligands or portion of the molecule that can bind to the surface, which explains the role in invasion and metastasis of cancer cells, because they can bind to the cells. Okay. Leukocyte adhesion deficiency is a rare condition. Now, this is a mutation which affects the Golgi located GDP focus transporter. So with the absence of this focus ligands for lectin leads to a marked decrease in neutrophil ruling. Neutrophil ruling will activate the neutrophils. So patients suffer life-threatening bacterial infection as well as psychomotor mental retardation. However, this can be easily treated by taking orally Focus. Focus is a deoxygenated deoxysugar or deoxyglucose. Another one is paroxysmal nocturnal hemoglobinuria, where there is hemoglobin in the urine, and it says nocturnal. So during the time you're sleeping, because of a slight drop in the plasma pH, so slight drop in the plasma pH. So if the plasma pH drop towards acidic side, which increases susceptibility to lysis by the complement system. The condition is due to acquisition in hematopoietic cells of the somatic mutations. So that's why we have this, what we call paroxysmal nocturnal hemoglobinuria. So again, these are very rare cases. Then you may have congenital muscular dystrophies. defect in the synthesis of glycans in the protein alpha dystroglycan. So the muscles here will suffer some dystrophy. Okay, continue. I will continue. I have had allergy, and that's why mucus is forming. Uh, I know I am allergic to shrimps, but yesterday was the shrimp was very tempting, so I ate some shrimps, and until now I suffered. Okay. Now, inclusion cell disease results from faulty targeting of the lysosomal enzymes. So this is, again, as we said, very rare cases. All you have to do is just mention them. I have not seen this inclusion cell disease. What is common is glycation of hemoglobin. Very common in the Philippines. And it says that 1 in every 3 Filipinos will develop diabetes mellitus because we have changed our way of eating. Okay. Now, all of these disorders are not really, you cannot even be able to see them or handle them. Okay. Glycans are involved in the binding of viruses, bacteria, and parasites to human cells. So these glycans can bind to specific proteins and other glycans. Now, this is taken advantage of by viruses, bacteria, and parasites. Sample your hemoglobin or your influenza virus A binds to the cell surface of a glycoprotein receptor molecule containing acetylnuraminic acid via hemagglutinin protein. The virus also contains an enzyme that acts on uraminic acid that plays a key role in allowing removal of newly synthesized progeny from infected cells. If this process is inhibited, the spread of the virus is markedly diminished. Inhibitors of this enzyme are the viral drugs like zanamivir and oseltamivir. They are now available in treating patients with influenza. But, we have already a flu vaccine which is quite effective. So unless, of course, you are not vaccinated for influenza. Now, there are several types of this influenza. And we are vaccinated by the most common type found in the Philippines. So there might be a few which cannot be covered by this influenza virus, but it's only a small number. Anyway, we have this zanamivir and oseltamivir to inhibit these particular enzymes. All right. Now, look at this. There are 16 types of this hemagglutinin and 9 types of neuraminidase, the enzyme that acts on uraminic acid. The avian flu is classified as H5N1. Hemagglutinin 5 and neuraminidase 1. Okay. HIV, virus type 1, cause of AIDS, attach itself to a glycoprotein GP120 and uses another surface glycoprotein GP41 to fuse with the host cell membrane. Antibodies to GP120 developed during the infection by HIV-1, and there has been interest in using the protein as a vaccine. The only problem here is that the virus can easily mutate. And if they can easily mutate, they escape the activity of the antibodies directed against it. That's because of this HIV, the virus easily mutates. I hope the coronavirus that we have will not immediately mutate and stay as is. Now, another bacteria cause peptic ulcer, the HP, Helicobacter pylori, cause peptic ulcers. They are present, the glycans are present on the surface of epithelial cells in the stomach. Is to establish a stable attachment to the stomach lining. Very similar to bacteria that cause also diarrhea. Similar also to the attachment of malarial parasite, Plasmodium falciparum. It is mediated by GPI, present on the surface of the parasite. Okay. All right. You have to excuse me because my throat is very itchy and I cannot be coughing into the microphone. So let's just end here. No? And Dr. Barito will meet you tomorrow. Okay? Welcome.
Thank you po.
Thank you, Doc.
Welcome.
Thank you, Doc. Love you, Doc. Have a nice day.
Welcome.
Get well, Doc.
Welcome.
Thank you, Doc.
Get well soon, Doc.
Welcome.
God bless, Doc.
Thank you.
Inom tubig, Doc.
Get well soon.
Yeah.